Good day, everyone. So this morning I'll be presenting some of my findings from my research project, which is looking at some of the factors which cause plum decline in the Western Cape. This project is under the leadership of Professor Teresa Coutinho. So this slide summarizes some of our main objectives. We are investigating some of the factors which we suspect to cause tree death in the Western Cape, namely the drought, ring nematodes, Phytophthora, as well as Pseudomonas syringae and Pseudomonas viridiflava, which causes bacterial canker. However, the focus of this talk will only be on the highlighted. So the overall aim of my thesis is to study the effect of drought and ring nematode stress combinations in predisposing plum trees to Pseudomonas syringae infection. However, the objective of this presentation is to investigate the association, if there is any, between ring nematodes and bacterial canker outbreaks. So we conducted surveys in 13 farms spanning 5 regions in the Western Cape. These are indicated by the red drop pins on the slide. These surveys were conducted a little over a year and a half, and there's one due in October. In the orchards themselves, we conducted tree assessments using the scoring matrix which appears on the slide. We also looked at the characteristic symptoms such as oozing of gamosis from the cankers, as well as dieback. So I'm just going to jump straight to the results. Logistic regression analyses were performed on the survey observations from the 13 farms. The first, ring nematodes and bacterial canker were treated as the dependent variables because those were the ones which we wanted to quantify. So the first model was the probability model for disease, which is disease infection, whereby a tree that is infected is as a result of drought, area of the farm, time of season, choice of rootstock, as well as ring nematode densities. So there were 395 observations included in the analyses. Of these observations, 222 were disease-free and 173 were disease-infected. We included a wild statistic to assess the significance of the association between the factors with disease infection. The wild statistic was significant, which means that the combined factors were associated with disease infection. We then determined the effect of each of the factors on the outcome of disease. We found that all but ring nematodes significantly affected the outcome of disease. Next, the probability model was for ring nematode, which was high infestation, which means that a high infestation is influenced by time of season, drought, area of the farm, as well as choice of rootstock. Again, the number of observations were the same as the bacterial canker model. However, 374 were not highly infested, and only 21 were highly infested. Again, the wild test was significant, which means that the combined factors are associated with high infestation. We then looked at the effect of these factors on ring nematode densities, and we found that area and drought were significantly contributed to high ring nematode densities, whereas the choice of rootstock as well as time of the season did not. So in conclusion, there is no association between bacterial canker and ring nematode densities in the 5 areas surveyed in the Western Cape, as we had initially hypothesized. Also, drought and area had the most significant effect on both disease outcome and ring nematode densities. What was interesting is that rootstock was only significant in the case of the disease outcome and not on ring nematode densities, as we had initially thought. With that, I'd like to thank our collaborators as well as our sponsors. Thank you.